Hello. Yes, sorry, it's been a while, but welcome back. I'm Dr. Ben Newman. I'm a coronavirus researcher, and let's get to your questions. Right, first question today is from Barb. How you doing? Yeah. Okay. Hi again. Can you please clarify what a gain of function test in labs that work on viruses is used for? Sure. Yeah. What does everyone think? Why does everyone think this is how the virus started? And the people in Congress saying it don't seem to be expert on the subject. They are not, but they believe it anyway. Yeah, which whatever. Yeah, I have beliefs on the new Taylor Swift album, but I am not an expert, and so I'm not going to be sharing those today. Yeah, it's fine. Yeah, except when you're making policy that determines basically whether the country survives and whether people survive. And yeah, basically, if you're going to play with fire, you ought to understand fire a little bit, I would say. So, yes, I picked up on the same things as you, I would say. Okay, so what's gain of function testing? This is a group of experiments that really are designed to figure out how things work. So, gain of function is something that usually gets diagnosed after you've done the experiment, but sometimes you can figure it out beforehand. For example, there was an influenza virus, H5N1, like one of the bird flu viruses. People grew it in ferrets. They then found out that after the bird flu virus passes through ferrets, it picks up one or more mutations that tend to make it more able to spread to humans. Because right now, bird flu, if you get it all the way in the bottom of your lungs, you can potentially get really sick. If you actually catch it, it's pretty bad. More than half of people die, if I remember right. But it's really hard to spread from one person to another, and you're also not likely to get it because you got to basically breathe in a lot or get really unlucky in that it gets to just the right cell at just the wrong time. So they found out, they put this into the ferrets, and all of a sudden they've got this now humanized version. And I believe two labs figured this out at the same time. They had some discussion with the governments, like, is this a thing that we should talk about? Is this a thing that we should put out there at all? And the argument in favor of not putting it out, that's pretty obvious. Just like, don't tell this, because if you put it out there, bad people will know it's out there. The argument in favor of telling it is that, one, ferrets are a pretty common model for flu. And so if you don't tell people, people are going to stumble onto this on their own without knowing that this is potentially what can happen. And that means that you have the risk of an actual outbreak connected to the science because of not telling. Also, it's important to know how things work, and information on its own isn't really good or bad. It's like what you do with it. The other counterpoint to the good people with guns versus bad people with guns kind of idea, with viruses, not guns, is that it takes really a lot of equipment and a lot of training to be able to actually do this. You can say, sitting in your living room, I've got a ferret right over there, and I got, I don't know, something, a chicken coughed out. I'm just going to, mash these two things together and, yeah, end the world. But you need the capability to sequence the virus. You'd need an egg culture system and a cell culture system to figure out the dose of the virus. Because if you get the dose wrong, you probably won't even infect the ferret. You need to make sure that it's exactly the right strain, because this probably doesn't work with all the strains. In fact, definitely doesn't work with all the strains. And you'd have to do the experiment enough times to know whether or not it's reproducible, like properly reproducible for everybody, everywhere, all the time. Yeah, in short, yeah, the idea is that somebody would want to do bioterrorism like this because they're really mad at somebody else. But one, once the cat's out of the bag, how are you going to stop it? And nobody's come up with anything in that department yet. Number two, if you're really mad at somebody, there are a lot of other ways you can get back at them that don't require putting yourself through grad school, getting that PhD, studying really hard for the thesis, working at a postdoc, maybe in something else, so you can secure some funding, because you're going to need a pretty good laboratory with pretty high containment to actually do these experiments. Securing the funding to do it, getting some collaborators in, because you're not going to want to take care of, the ferret feeding and everything else on your own. And basically, you get to a point where I'd like to say you could probably spot the skull-shaped island from space if this was actually going on. It's not, it's not a low resource or fast thing. And so, in general, this is more a thing that people worry about than anything that's actually particularly dangerous. But at the same time, it is a concern, and you know what? There are ethics boards, there are biosafety boards that have oversight over everything that gets done in the lab, and this is the stuff they should be thinking about and the stuff they do think about. And so an experiment like that, at the time, wasn't being done as gain of function. People weren't saying, I want to see if I can make, a human-killing version of bird flu. They were doing it to try to actually study the virus. How does it grow? And ferrets are a good model. They have similar lungs, and they take to influenza similarly to humans for some other strains. Other examples of gain-of-function research are, say you've got a bat coronavirus, like all the ones that lead up to original SARS or SARS-CoV-2, new and improved. But say, like is always the case, these are viruses that haven't ever been grown and can't actually be grown because they probably have some problems. What you can do is you can take the spike out of one of those viruses and put it into another virus to see, a related virus, to see, all right, how does this thing work? Will it grow? Can I study it? Are there any differences in the body of the thing? And you can do the converse as well. You can take a virus that isn't able to get into cells, and you can say, well, what if it could get into cells? So you can put on a regular spike, and you say, okay, now, is this a thing that's going to cause more disease, less disease, no disease, or is it going to be broken in some other way because there's some other incompatibility in there? This is basically a way that you can learn what the different parts of a new variant or a new strain do. And it's only really gain of function if you happen to make a virus that seems as though it grows well and can cause more disease. Or cause disease in a different organism than the original. And you kind of don't always know that until you've done the experiment. So a lot of things that started out as probably not gain of function turned into it, then get shut down because we say, nope, that looks as though you may have gained a function, and don't do that. We don't even want to take a chance. So that's basically the story. And yeah, this stuff's been done various places, various times, probably more in America than anywhere else. But yeah, whatever. It's neither here nor there, right? There you go. So hope that answers at least some of your question. Thanks very much. This has been Ask Dr. Ben.